So my research is based on Alzheimer's disease. I wasn't sure if you know what Alzheimer's disease is, so I put a little bit of introduction. It's a form of dementia, and it's the most common form of dementia in the world, and it's a progressive neurodegenerative disorder. What this means is it's neurodegenerative, so it's caused by brain cells dying off, and it's progressive, so as the disease continues, more and more brain cells die. And this is what causes the symptoms of Alzheimer's disease, and these are things like memory loss, which you've already heard about, but other things like personality changes and depression and psychosis, confusion. And as the disease gets more and more advanced, people generally lose their ability to perform normal daily tasks, and that includes things like how to eat, how to walk, how to write a letter. And if we look at the brain of Alzheimer's patients, we can actually see this neuronal loss. That's how dramatic it is. So over here we've got a healthy brain. This is a section of the brain, so it's like if you swipe through my head that way. And this is a normal healthy brain. This is the brain of an Alzheimer's disease patient, and you can see it's a lot smaller. This dashed red line is the size of the healthy brain section compared to the Alzheimer's. And the shrinkage in the brain is simply just because so many brain cells have died. And you can see that there's big holes in the brain, and it's specifically important in this area here, because this is where the hippocampus is. And the hippocampus is where all your memories are stored. And this is the first part to degenerate in Alzheimer's disease, which is why we get symptoms such as memory loss. So you can see if we've lost that section of the brain, we've lost that function. If we were to stain up brain tissue from Alzheimer's patients and look at it under a microscope, you can see the difference there as well. So again, over here we've got an example of some healthy brain tissue, and this is Alzheimer's brain tissue. And what we see are two main hallmarks of the disease, and these are amyloid plaques and hyperphosphorylated tangles. And these plaques are outside of the cells, whereas the tangles are inside of the cells. And what they both are is aggregated forms of a normal cellular protein. So a protein is synthesized, its primary structure is the sequence of amino acids, it then folds into its secondary structure, which is alpha helix or a beta pleated sheet, and then finally folds into its tertiary structure. What's happened here is that it's misfolded, so its tertiary structure is incorrect, and that's caused it to aggregate together, forming these clumps. So where do these come from?
You might want to use the mouse, it might be easier if you just click it.
So the amyloid plaques are composed of a peptide called amyloid beta, and this is synthesized from a much longer precursor protein called amyloid protein, and this is APP up here, or APP is its acronym. APP is, like all proteins, it's synthesized in the cell, it's then post-translationally processed, and what this means is it has extra sugar stuck on it, and then it's cleaved by two enzymes. And it's when it's cut by these two enzymes that we get this fragment in the middle, the amyloid beta release. And this is the amyloid beta monomer. This happens all the time. It's happening in your brain, it's happening in my brain, and it has a normal physiological function. We don't know what this is, but we know that it has a normal function in a normal brain. However, for some reason, and this is the bit we don't understand properly, in Alzheimer's disease patients, this amyloid beta monomer misfolds, and too much of it builds up, and in this misfolded conformation, it sticks together to form these amyloid beta oligomers. And although this monomer in its normal form has a normal function, in this misfolded and aggregated form, it's toxic. And it's these oligomers which can kill surrounding brain cells. And these oligomers then further aggregate, forming the plaques which you saw on the previous picture. We can use the mouse system. And you can see that again here. So we've got normal brain cells with normal monomeric A beta, this has a normal function. However, if it aggregates, it forms these toxic structures which then kill the surrounding brain cells. And the hypothesis, which has been the most widely accepted hypothesis for the last 10-15 years, is called the amyloid cascade hypothesis. And this proposes that it is this amyloid beta which is the primary cause of Alzheimer's. So that means that amyloid beta is the thing which starts off all the toxicity. Because remember I showed you in the previous picture there was another aggregate that was the hyperphosphorylated tangles. We think that this happens first and then those happen downstream. So a lot of the research efforts into trying to find drugs for Alzheimer's disease has focused on things along this pathway. So what we can try and do is we can try and inhibit amyloid beta generation, we can try and prevent it from folding in this way, and we can try and stop these things being toxic to the cells. And those are all strategies that the drugs companies have been trying for quite some time. So how do we go about designing a drug for Alzheimer's, or for any disease in actual fact? What we do is we start off with very basic research, which we'll do maybe in cells in addition in a lab, maybe with just single enzymes in a test tube. And in that we're trying to establish what cellular pathways are involved in the disease, and possibly how these can go wrong in the disease. So once we've looked at this in a cell line or in a test tube, we've seen that this is true in the cell. We now need to know, is this true in a more complex organism? So we use a model of the disease, and that's quite often a mouse or a rat, or sometimes even a fly or a worm, which often it's the elements. If we find that this is true in the mouse as the same as it was in the cells, we then look at what parts of that pathway we can alter to try and prevent the disease state from happening. So we'll try and find a drug. We will do that again, we'll do that partly in mouse models, but we'll probably go back to cell lines for that, because we'll make a whole range of molecules. We'll look at what they can bind, how well they can bind, can they inhibit a particular enzyme in a particular pathway. So once we've started in cells, we identify the pathways, we see if the same is true in another organism, such as mouse. We then try and alter those pathways to make a drug, and at this point we think we've maybe got a drug that works. What we need to do now is we need to test it. We need to test two things before we can really start. We need to know, is it safe? So does it have any toxic side effects? And how much do we need to give what dose? And for that we need to go back to the disease model of the animal. So what we'll do is we'll take our Alzheimer's disease mice and we will give them different doses of the compound, and we'll check for any adverse side effects. And we'll also check how much of the drug actually gets into their brain, so we know how much to give them, whether the enzyme that we're trying to inhibit or activate is actually inhibited or activated. And once we've done all that, then we can finally see, does the drug actually work? And for that, again, we have to start it off in animal models of the disease. So we'll take a lot of animals with Alzheimer's disease, we'll give them the drug over a period of time, and we'll measure whether this improves the disease progress. So does it reduce their amyloid plaques? Does it improve their memory? So to do this, obviously we need an animal model of Alzheimer's disease. Mice are a very good. Now, mice are a very good animal model to use for several reasons. They're well characterised, and what that means is that we've done a lot of experiments on mice over the years. We know very well their genetics, we know very well how they behave, so we know what to expect from them normally. They breed very fast, so it's easy for us to get lots and lots of mice to work with, because we need a lot of mice to draw valid conclusions from our work. And they're relatively similar to humans. Now that might sound a bit silly, but when I say they're relatively similar to humans, I mean compared to, for example, flies. So in Alzheimer's disease, mice are quite good because they have a brain structure similar to humans, they have a hippocampus, they have the same organs as humans. However, we do have a slight problem, and that is that mice don't naturally get Alzheimer's disease. So how do we alter the mice so that we can model Alzheimer's disease? Well, the reason that they don't naturally get Alzheimer's disease is that their amyloid precursor protein, which was the protein which forms the amyloid beta and the amyloid beta plaques, actually has a very slightly different primary structure to ours, so it has a different sequence of amino acids around the amyloid beta part. And what this means is their amyloid beta doesn't aggregate, and this prevents them from getting the disease. So to try and, well, to not solve the problem, it's not really a problem for the mouse, to try and overcome this, what we've done is we've genetically altered them. So we've taken out the mouse APP gene and we've put in the human APP gene. And we've actually gone one step further. We've put in a mutant human APP gene, and it's called APP Swedish. And this is because it comes from a family from Sweden, and it accelerates the formation of Alzheimer's disease. So much more amyloid beta is produced from this APP. And the people who have this mutation get Alzheimer's disease much earlier. So if we put this gene in the mouse gets Alzheimer's disease much earlier, and that means that we can start working on them earlier, and we have more pathology to look at. So what I do is, my job is to look after these mice and maintain them before we use them for experiments. Part of my job, I should say. What we need to do, rather than genetically alter a mouse every time we want to use it, we take a mouse that we've already genetically altered, we've already tested, we know it's got the APP Swedish gene in it, and then we maintain our transgenic stock just by breeding them. So we'll breed a transgenic mouse with a normal mouse. That gives us lots of mouse babies, probably more than that, but it wouldn't fit on the slide. Only now we have a problem. We have lots of mouse babies. Some of them are transgenic and some of them are not. And we need to know which is which, because we need to know when we do our experiments, we're going to need to do them on transgenic mice, but also on control mice, so that we know what we're comparing to. And that's where the PCR that we're doing today comes in. The way we determine which mouse is which is we take a small sample from the mouse. So we had today tail tips, which you can have a look at in the tube, or we might take an ear punch, and we extract all the mouse DNA from that sample, and then we do PCR to look for the APP gene. And because we've replaced the mouse APP with human APP, if we design our primers carefully, then we can amplify just human APP. So that means if the mouse is transgenic, we extract the DNA, we do a PCR on the DNA, we put in primers for the human APP. If the human APP is there, the primers bind, the PCR reaction happens, and we get a PCR product. However, if there's no APP gene, if the mouse is not transgenic, there's nothing for the primers to bind to, so we get no PCR reaction and we get no PCR product. However, as I explained this morning, I don't know how many of you were here, if we get no PCR product, that could actually be for two different reasons. It could be either because the mouse is wild-type mouse, who's not got the APP gene, or it could be because our PCR reaction didn't work for some reason. So we need to be able to distinguish those two. And for that we use a control gene. So we extract the DNA from the mouse, we have all the mouse DNA, including the human APP gene, and then we do the PCR reaction, and we do simultaneously 2 PCR reactions in the same tube, which is what we set up earlier today. We set up a PCR reaction for APP, that will tell us if the mouse is transgenic, but we also set up a PCR reaction for just a normal mouse gene. So every single mouse should have this gene in it, so every single PCR reaction should come up with a band. In the APP, the APP mice, the APP primers bind to the DNA, and we get a polymerase PCR reaction and a PCR product. And also the normal mouse primers bind to the DNA, so we get a PCR reaction for the normal mouse DNA and a PCR product. However, in the non-transgenic mice, the APP primers, they have nothing to bind. So they don't bind to the DNA, so we get no PCR reaction. However, the normal mouse primers, there's still normal mouse DNA in there. So they bind to the mouse DNA, and we get a PCR reaction, and we get a PCR product. And so you can see from the APP transgenic mouse, we get a long APP product. And we've designed these primers so that the products we get are different sizes, which means we can very easily distinguish them on the gel. So we get a long APP product. We also get a short normal mouse product. Whereas in the non-transgenic mice, we don't get any APP product because there's no APP in there, but we get the short generic mouse gene product. So that tells us that the PCR reaction has worked. And I've put in an example. Hopefully your PCR reactions will look something like this. This is an example of some PCR I did on some of my mice. And the first thing we need to do when we look at this gel is to look at the controls, because the controls will tell us whether the experiment has worked, and if the controls haven't worked, then we can't conclude anything from the experiment. So firstly, today we did a negative control, and that was a PCR reaction that we didn't put any DNA in. And that's very, very important, because it's very easy to contaminate a PCR. So if we get maybe some of your DNA in there by accident when you're doing it for the mice, you've got human APP because you're a human. So you'd amplify up a product and you'd get a band, and you'd think your mouse was transgenic, but actually that was just your DNA. So to check that we haven't contaminated any of our reactions, we run a sample in which we put no DNA. So we expect no PCR product. If we get something in this, we have to discard all the gel, we have to do the experiment again. So this one was okay, you can see there's nothing there. And the other two controls are a positive control for wild type and a positive control for transgenic. And these are DNA from mice that I've had before that I already know are wild type or are transgenic. And you can see we've got 2 different size bands, so they run at 2 different sizes on the gel. The big APP PCR product is bigger, so it runs more slowly because it's harder for it to get through the gaps in the gel. And the smaller mouse gene runs more quickly because it's smaller, so it can much more easily get through all the spaces and the gaps in the gel. And this shows us that both the PCR reactions for the wild type and the transgenic mice should have worked properly. So So for the wild type mice, we've got what we expected, just 1 band. For the transgenic mice, we've got what we expected, a band for APP and a band for the normal mouse. You can't see that very well, but it is there. And so this tells us that our whole experiment has worked how we think it should. So we can then use this to work out what all of our mice are. So I've highlighted the transgenic. It should be very obvious to you which are the transgenic mice. And I've highlighted them. Another thing that we might use transgenic mice for, I drew you out a diagram earlier of the stages of drug discovery, but another thing that we use transgenic mice for is in the very initial stages of research. So when the APP Swedish gene was first found, we knew it caused Alzheimer's disease, but we weren't sure how. And as we did some research in cells, we found out that we think it causes more amyloid beta production. And to confirm that and to try and look at further phenotypes that it might cause, what we did was we put it into mice. And we then looked at the mice to see how they were different from normal mice. One of the very important things that you can do when you first created a mouse line is to look whether it affects their development, whether they're born as normally as they should be. And an easy way to do that is to look at the ratio of transgenic and non-transgenic mice. So we know what we've crossed here. We took a heterozygous transgenic mouse, so that's a mouse with 1 human APP gene that we genetically inserted, and 1 normal APP gene that was normally there in the mouse. And we crossed it with a normal mouse. So he had a normal APP gene and another normal APP gene. He was a homozygote. So we've got a cross like that, transgenic and normal, normal and normal. And we can look at the expected ratio of the offspring. And if we do that, we can see that when we do this genetic cross, we'd expect 50% of our mice offspring to be transgenic and 50% of them to be wild type. So if we were to do that cross and we were to find that maybe only 10% of our mice were transgenic, whereas 90% of them were wild type, that might tell us that this gene was causing some problems in development. So the mice that were getting the transgenic gene are not surviving long enough to be born, for example. So that's a very simple first test you can do. So we can do that on these mice here. This is what we'd expect, 50% transgenic, 50% wild type. And we can look at what we actually get out of the 10 mice. So we get 60% transgenic and 40% wild type. And that's pretty close to what we'd expect. From only 10 mice, which is not a huge number, that's pretty much 50-50. So that tells us that these mice are developing normally. And that's what we find. If we were to do lots and lots and lots of mice over a long time, we find the numbers get closer to 50-50. So that tells us that our mice are normal. And that's it. Hopefully your gels will be as pretty as that. But that is my last one. So have you got any questions?
How do you insert the Swedish APP?
The Swedish APP gene. It was actually done a long time ago in the early 2000s. What they did was they took a virus construct which had the human APP gene and then the ends of it were homologous to the mouse DNA. So you could take an egg cell of a mouse and then in a petri dish you can inject into the nucleus the construct, the viral construct that you've made. And the bits that are homologous, so that's basically the same as the mouse chromosome either side of the APP gene, will line up with the mouse chromosome, and then there are enzymes called recombinases, and they will swap. Where there's 2 bits of DNA lining up, they'll swap the middle bit over. So what happens is the mouse bit gets swapped in the human bit. So you end up with mouse chromosome, human gene, mouse chromosome, where you had mouse before, and then that can be implanted back into a mouse. So it sounds very simple, but it's really not. It's a lot of work.
If the mouse gene, like if we know that it doesn't produce Alzheimer's, so then, like, why can't we use with anti-human, like, see what that would do?
That would be a very big ethical problem.
I mean, there must be ways you can do it without actually inserting it straight into actual humans.
Well, there have been studies done where you alter the human APP and prevent it aggregating. And there are actually naturally occurring mutations in the human APP which increase the aggregation, and these increase chances of developing Alzheimer's. So we know that this would help, however, in terms of finding a drug for the disease. It's not something we can actually do because we'd have to go right into the cells, we'd have to alter the DNA from very early on in a person's life. And that's not really feasible. One of the things that makes Alzheimer's disease incredibly difficult to treat is that it's very, very slow progressive disease. So if you were to lose one brain cell, you wouldn't notice the difference. If you were to lose 2 brain cells, you wouldn't notice the difference. So you have to lose quite a lot of brain cells before you start getting symptoms of Alzheimer's disease. And by the time you started getting symptoms, firstly that means you've lost a lot of brain cells. Once the brain cells are gone, we can't put them back. So we need to start treating the disease very, very early on before we get the symptoms. Only we can't predict at the moment who's going to get Alzheimer's disease. So we can't start doing that yet, which is one of the reasons it's been so difficult to find a cure.